Alright, hi everybody, this is Lilith and I'm one of your IAs for BIPN 160. In this video, I'll do a review of Lecture 11, just the first half up until PVH, and then I'll do the second video covering part 2. Okay, so in Lecture 11, we started to talk about the hypothalamus and the structures within it and the structures outside of it, and how many homeostatic functions are taking place in our bodies. So to begin with the hypothalamus, the functions of hypothalamus are right over here. It regulates appetite, regulates weight, regulates water balances in the body, sleep cycles, sexual drive, and aggression. Okay, the hypothalamus is located right underneath the thalamus, right here. So this big blob is the thalamus, and right below it is the hypothalamus. Structures around the hypothalamus, you will notice that this little structure over here is the optic chiasm, and this makes sense because our eyes are right about here, and so the optic nerve goes all the way to here, and that's where it crosses. Right behind the optic chiasm, we have the pituitary gland, and the pituitary gland is very connected with the hypothalamus, and it listens to the hormones that the hypothalamus releases and sends it to. Okay, and we will get to talking about the pituitary gland in just a little bit. Okay, so the hypothalamus itself, it's very small, however it has many nuclei, and it also has many inputs. So the nuclei that you definitely want to memorize, and definitely want to memorize the functions of, are right here. I cropped them out from that list Dr. Root gave you, and so these bolded ones are the important ones that you should definitely know. We'll go more detailed in this video about the arcuate nucleus, and probably that's it. And then the rest, most of these we'll go over in the next part 2 of Lecture 11. Okay, but just make sure to memorize these. The hypothalamus also has many inputs from different areas of the brain. It's very interactive. So it has inputs from the amygdala. A lot of amygdala is involved in a lot of homeostatic functions. Different cortical areas also input into the hypothalamus, the brainstem and spinal cord, and also, of course, the retina, because signals directly from our retinas or from our eyes are what's going to help us with the circadian rhythms, right, that we will discuss in Lecture 12. Okay, so know the inputs to the hypothalamus also. So that was kind of an overview to the hypothalamus. Then we went on to talk about the pituitary gland. So here's an overview of pituitary. I took this picture from online because the one that was from the book might have been a little provocative, so we'll just go ahead and look at this one instead. There's more information on here than you need to know for this class, but I went ahead and written out that this part right here is the anterior side, and this part right here is the posterior side. This is the whole pituitary gland, and we have an anterior portion or chamber, and then we have the posterior pituitary. They differ in the types of cells that they have and the way they are structured. So the anterior pituitary contains the parvocellular neurosecretory system, I think that's what it was called, and that name, parvocellular neurosecretory system, refers to these neurons coming down from the hypothalamus and releasing hormones into this portal vein. So in this, I guess, vein and artery network, okay, then in the blood, those hormones flow into the anterior pituitary, and they activate cells that are in the anterior pituitary. Then those cells are able to release other hormones that are put into the blood through this venule, or put into the body. Okay, I'll explain that a little more in the next picture. So that's the anterior pituitary, and the posterior pituitary contains the magnocellular neurosecretory system. And this system contains these much larger neurons. It doesn't show the difference in this picture, but these magnocellular ones are supposed to be a lot bigger than these parvocellular ones. So the magnocellular neurons, instead of being just like the anterior pituitary neurons, they reach all the way down into the posterior pituitary, then release their hormones right here instead of releasing them right here. Okay, so that's the difference. I'm not sure if you need to know that detail, but it'll help you for other classes also to visualize that. Okay, so then I pulled out these photos to talk about the anterior and posterior pituitary separately. So to localize or to figure out where we are right now, see this little nub? That's the optic chiasm, meaning that this is the anterior pituitary. We have our neurons coming down from the hypothalamus. We have our huge portal system right here. And what I'd like you to understand in this picture is these neurons up here release hormones into this portal system, and those hormones are called stimulating hormones. Okay, and why are they called stimulating hormones? Because these hormones will be put into this portal system and they will flow into the anterior pituitary. And you see these cells in the anterior pituitary? These cells contain receptors for those stimulating hormones. And those stimulating hormones will activate different cells in the pituitary, and those different cells will be releasing other hormones that will be placed into the portal again, and now they will travel through this portal out into the body. Okay, for reference, this picture does a good job. These circles right here, these are stimulating hormones that are released by the hypothalamus, and the triangles here are hormones that are produced by the anterior pituitary itself. Okay, here's a list of the hormones that are produced by the anterior pituitary. So FSH, LH are for, we can call them reproductive hormones. TSH is the hormone for stimulating the thyroid. I just realized I made a mistake. When I said that these hormones released here are stimulating hormones, I said the wrong word. They're called releasing hormones. So sorry. Okay, I'll write it here. Releasing hormones. Okay. And then we have ACTH. That's a hormone that activates cells in the adrenal cortex, prolactin, mammary glands. MSH, we don't really talk about this, so you don't have to worry. And then growth hormone activates tissues that are producing other factors for growth. So basic idea, from the hypothalamus, there are neurons that release releasing hormones, and then those releasing hormones come to the anterior pituitary, activate cells, then those cells of the anterior pituitary release their own hormones, which are these guys right here. Okay, then these hormones go into different organs, for example, testes, ovaries, thyroid, or adrenal cortex, and they activate those organs to produce whatever they produce. Okay, so that was the anterior pituitary, and here on the side I have the posterior pituitary. Now, what's different about the posterior pituitary, again, I mentioned previously, is that these neurons don't stop where the pituitary begins. They actually travel all the way down to the posterior pituitary, and they release the hormones that they are making right into here, and those are the hormones that go out into the body. Okay, so in the posterior pituitary, the hormones that exit the posterior pituitary are the hormones that are produced by the hypothalamus itself. And those two hormones are ADH, otherwise known vasopressin, and oxytocin. Okay, we'll talk about ADH or vasopressin in just a little. It acts on the kidney tubules, or the functional units of kidneys, and it allows for us to keep more water inside of our body and not release as much water. Okay. One full, hormonal axis that Dr. Ru talked about, and he asked you a question on it during lecture also, is the hypothalamic pituitary adrenal axis, short HPA. Definitely this one you should look over, memorize. So what happens here is corticotropin releasing hormone, CRH, is released from the hypothalamus over here. Then the CRH activates cells in the pituitary, in the anterior pituitary, and then the anterior pituitary releases ACTH, adrenocorticotropic hormone, and then ACTH goes all the way to the adrenal glands that are sitting right on top of our kidneys. These are the adrenal glands on top. They sit like a hat, and ACTH activates the adrenal gland to produce cortisol. And cortisol is the stress hormone, or stress molecule, and it happens because of environmental stress or just internal stress. Okay, so I definitely recommend going over that and memorizing that axis. You should also pay attention to, I think Dr. Root mentioned 2 other axes. So one was CRH, the other one may have been TSH, and I'm forgetting the other one, possibly prolactin. So make sure to look at those also, because he might ask you a question, like an axis question, but it might not be HPA. It might be, for example, TSH. Okay. Okay, now that we have a good overview, let's talk about the first function of hypothalamus, which is the regulation of thirst. So in the hypothalamus, we have 2 nuclei or organs that regulate thirst. The first one is supraoptic nucleus, and the second one is subfornical organ, or SFO for short. What's important for you to know is that both of these brain areas or hypothalamic areas respond to higher blood osmolarity. Okay, so when I drink a soda or I drink a sugary coffee, the osmolarity of my blood obviously increases because I'm not drinking just, normal water filled with electrolytes or whatnot. I'm just drinking something that is going to increase my blood osmolarity. So when that happens, the supraoptic nucleus becomes activated. And how does it become activated? The neurons in this nucleus are able to sense the high osmolarity by either shrinking or puffing up. So here, the first scenario is us having plus 30 milli OSM. I forgot how you say OSM, but it's a unit of osmolarity. So when we have increased osmolarity, these neurons release a lot of the water that they have inside of them, right? Because they have like intracellular fluid. So some of the water comes out into the environment of that neuron because they're trying to decrease the osmolarity of their environment. And by doing so, they shrink. And when they shrink, they activate mechanoreceptors on the neuron. And once those mechanoreceptors are activated, the neuron starts to fire. So the neuron becomes activated. And when that neuron is activated, then the brain can tell, right, from the supraoptic nucleus, can tell that, hey, there is high osmolarity in our blood right now, so let's release vasopressin, or ADH, antidiuretic hormone. Antidiuretic hormone will stop us from going to the bathroom so often, like going to urinate, so that water will be retained in our blood more, and the osmolarity of our blood will decrease. In this case, in this scenario, there is actually decreased osmolarity. For example, if I just drink a lot of water throughout the day and no minerals whatsoever, which is also bad, so these are like two extremes, then that extra water will enter the neurons and puff them up, and when they are puffed up, the mechanoreceptors will not be activated, so the neuron will not fire at all. And if it's not firing, then we will not release vasopressin, otherwise known ADH, because if we already have a lot of water in our blood, we don't want to retain any more water. So we will not release vasopressin. So that's how supraoptic nucleus works. The subfornical organ, or the SFO, also responds to high blood osmolarity, but it works in a little bit of a different way. I don't think we talked about the exact mechanism of how it works, but all you should really know is that the SFO drives drinking behavior, or wanting to drink a lot of water. And this behavior is shown through this experiment right here, where I think they used channel rhodopsin and they activated the SFO, and you can see that when there is light to activate the channel rhodopsin in this time frame, there is so much more activity going on here in this neuron, and the water consumption or drinking response drastically increases when there is light. So when we activate this area, then this drives water drinking behavior. Okay. Now, after this research experiment, I guess, they also found that SFO contains two types of neurons: inhibitory neurons and excitatory neurons. As we learned in this class, usually GABAergic neurons, neurons that release the neurotransmitter GABA, are inhibitory neurons. And the ones that release glutamine, glutamatergic neurons, they are excitatory. So GABAergic neurons will actually inhibit drinking behavior, and glutamatergic neurons will excite or activate drinking behavior. Okay, and I didn't go over the experiments pertaining to these two, but it's a pretty simple concept. Hopefully you guys already know. If we activate the GABAergic neurons, then the mice would stop drinking water because this already is inhibitory. So if we activate that inhibitory neuron or neurons, then the mouse will stop drinking water. And then if we activate this, then the mouse just drinks water like crazy. Also, if we inhibit this, the mouse will stop drinking water. Okay, so just, if you have questions, please email or come to office hours and I can talk more about those, but I feel like kind of there's a theme in the class of, inhibiting and activating different areas and seeing what happens, so you should be familiar with that already. Okay, so done with thirst. The appetite regulation is the next topic, or the next function of the hypothalamus, so let's get into it. So there's a lot of different factors included in appetite regulation, just like you saw on the slides: the stomach, the brain, leptin being produced from fat cells, and sorry, my friend said hey. I forgot my train of, I forgot what I was saying. Oh yes, appetite regulation is regulated by a lot of different parts in our body, but for now we will be talking about the hypothalamus only. So this picture is basically summarizing how food intake is being regulated. So to my understanding, the key points to take away for you here are: whenever we have an energy deficit, meaning that we haven't eaten for a while, this will produce a negative valence. We will be hangry, right? We will be cranky. And then this negative valence will activate food intake. So we will go ahead and get some food, seek food if you're a mouse. And then whenever you do get the food, this will come back and inhibit this negative valence. We'll be happy after you get some food. And then we kind of skipped over the positive valence, so we'll just not go over it. The aversive visceral feeling, I guess, is when you eat something that is either bitter or you just don't like it. So whenever that is activated, then you inhibit food intake and you just don't want to eat any more of that food. Okay, that's kind of the gist of what Dr. Root talked about for those. These 3 main points are very important. You should definitely memorize these. Okay, let me zoom in. So, the desire to eat leads to food seeking, and that statement is found from the AGRP neurons in the arcuate nucleus. So when we activate AGRP neurons in the arcuate nucleus of the hypothalamus, the mice have a bigger desire to eat food or go seek food. Consumption of food is regulated in the neurons of the lateral hypothalamus. And then terminating eating or preventing it, so like not wanting to eat it because it tastes bad or just feeling full and stopping to eat, has to do with the CGRP neurons in the parabrachial nucleus. Okay, now let's talk about the arcuate nucleus first, and then we'll go on to the other ones. So the arcuate nucleus is right here, and some key things that you should know about it is it contains 2 types of neurons. It contains AGRP neurons and POMC neurons. AGRP neurons promote appetite, they promote you wanting to eat food, and POMC neurons decrease appetite. Okay, the AGRP neurons project to the paraventricular hypothalamus, which is, where is it? Right here. Okay, and we will talk, and they inhibit it. So the AGRP inhibits the PVH, and we will get back to that in just a little bit. This study over here is showing how the ablation of AGRP and POMC affect body weight. So up top over here, we are inhibiting AGRP. And so you see when this is just regular mice feeding, but when we inhibit AGRP, the mice will start to lose weight because AGRP initially promoted eating. But if we inhibit it, the mouse stops eating. And over here, if POMC is inhibited, comparing to the regular mouse, as in like the control, these mice start to gain weight as soon as POMC is inhibited. Remember that POMC decreased appetite, but if we inhibit that, then the mouse will start eating way, way more. Okay, and then these graphs show an interesting finding that we will talk about later. The fact that during these experiments, the blood glucose is not changing. I think that's what it's saying, at least over here. Well, now I can't remember if Dr. talked about this part of the graph. Let's just skip it for now, and then I'll look through the lecture again, and I'll include it in my next video if he talked about it. But for now, remember that POMC decreases appetite, AGRP promotes appetite. In this study, they talked about how they activated AGRP with channel rhodopsin, and this is again a very simple study, right? If you activate the AGRP neurons by channel rhodopsin, then here where it is stimulated by light, you see that more of the pellets are being eaten by the mice, and then as soon as you stop stimulating, they just calm down again. But, that's crazy. As soon as you stimulate, they just start eating food, and then they don't anymore. Okay. Before I move on, remember that active AGRP neurons allow the mouse to feel negative valence. meaning when these are active, your body realizes that you are hungry. And this gives you a feeling of being hangry, right? So you start to seek food. This study over here showed us exactly when AGRP is active. Is it active when the mouse is starved, or is it active when the mouse is fed? And so, long story short, they figured out that the AGRP is active right up until food is given. So when the mouse starts eating, AGRP calms down. It's inhibited. And then when food is taken away, the mouse is hungry again. So the AGRP starts to be active again. And then it calms down, and then active again, then it calms down. And then, finally, when the mouse has had enough food through these cycles, the AGRP is just completely being inhibited, because AGRP is active only when the mouse is hungry or seeking food, and it is inhibited when the mouse is fed. Okay. Then they did another study where they tried to think, well, is the inhibition of the AGRP neurons coming from the fact that they recognize the levels of glucose in the blood? And the answer was no, because the inhibition of AGRP happened too fast, right? There was no time for the glucose to even enter those neurons. However, they realized that the possible reason to how AGRP becomes inhibited is the expectation of food or seeing food. For example, when the AGRP is active and the mice are presented with food, then AGRP gets inhibited and then normalizes. And the same thing happens here. AGRP is active, and then the mouse is given false food. So for a quick moment, this dips because the mouse thinks, oh yay, that's food. But then when it realizes it's not food, AGRP is active again. Okay, so expectation of food or the visual of food is what inhibits AGRP. And the last topic that I want to cover in this video, because I have to go, I think, do another study session soon, but remember when we talked about how AGRP projects to the HVP up right here? So AGRP projects to the paraventricular hypothalamus by inhibiting it. So this was a study that looked at the inhibition of HVP, and they found that the inhibition of HVP causes feeding, and this further confirmed that AGRP drives food-seeking behavior. Because if you think about it, if AGRP inhibits PVH, right, and this causes eating, right, then the activation of AGRP, right, AGRP must have been activated to inhibit PVH. So if the manual inhibition of PVH activates eating, then we know that activation of AGRP must also drive eating or food-seeking behavior. Okay. Hopefully that made sense. I'll come back with part 2, hopefully tonight, of lecture 11. And I'm hoping lecture 12 will be on Sunday. Okay? Please email me if you have any questions. This will be posted in the next 30 minutes or so. Okay, thank you.